Fine. So yes, in our previous class we have dealt up till completion of prokaryotic cell, genetic material, cell envelope, non-membrane bound organelles like ribosomes, inclusion bodies, motile and non-motile structures. Till there it is done. Now we will look on to different sizes and shapes of the cell. If you look into different shapes and sizes, first of prokaryotic cell, circular or spherical shaped bacteria are known as what? Cocci. Singular is coccus. Rod shaped bacteria are bacilli. Singular is bacillus. Spiral shaped spirillum. Plural is spirilla. Vibrio. Comma shaped bacteria are what? Vibrio. Okay, and singular is vibrium. Okay, these are the terms that you have to be noted with. Amongst this, which is the most pathogenic bacteria? Most pathogenic bacteria. Bacillus is the most pathogenic bacteria are of bacilli in shape. Now coming to different sizes of bacteria, sizes of different cells, not just bacteria, sizes of different cells. From this you may get like diagram based questions or statement based, any type, match the following, you can get any of these forms. Eukaryotic cell will be around 10 to 20 micrometers in diameter. Typical bacteria, any bacteria can be of 1 to 2 micrometers in diameter. PPLO, which is known as pleuropneumonia like bacteria, which is a type of mycoplasma, right? PPLO is what? Pleuropneumonia like organisms. This is one type of mycoplasma. Which varies in its size around 0.1 micrometers in diameter. Next is viruses, which is around 0.02 to 0.2 micrometers in diameter. Okay. Wherever you feel like it is an add-on point, till now it is not there either in your NCERT or in your running notes, you can add on that, because again it will be like a fresh wording to you if you are not taking it off. Okay. So here we have learnt on different shapes of bacteria, of which most pathogenic type are of bacillus. Next we have looked on different sizes. Eukaryotic cell is of what size? Approximately 10 to 20 micrometers. Bacteria 1 to 2 micrometers, PPLO 0.1 micrometer in diameter, and viruses is around 0.02 to 0.2 micrometers. Amongst this, the smallest is PPLO. Mycoplasma is the smallest living cell. Okay. Next. Yesterday we have learnt on gram staining, but we didn't learn the entire procedure, right? So gram staining, which is proposed by Christian Gram, what did he say? In our NCERT the statement is like, whether it is gram positive or gram negative, cell envelope will decide. Okay. Gram positive and gram negative bacteria is based on the type of cell envelope. More preferably, more precisely if we have to say, it is majorly dependent on cell wall. Here in this diagram, I would like to show you the differences in the cell wall composition. Can you see here, my everybody? The cell wall composition. In gram positive bacteria, if you observe gram positive, 80% of peptidoglycan is present and just 20% of polysaccharides and lipids are present. Whereas in gram negative bacteria, if you observe, just 20% of peptidoglycan, 80% of polysaccharides and unsaturated lipids. Polysaccharides and unsaturated lipids are present. Got it? This is about gram positive and gram negative bacteria. Now, what is this staining procedure? How you will get to know about gram positive, whether this bacteria is gram positive or gram negative? What is Christian Graham's experiment? He has taken two bacteria in two different petri dishes. Let us suppose, okay, listen ma first. Two bacteria taken in two separate dishes, petri dishes. Let us suppose that. Let us suppose this as gram positive, we do not know, but let us suppose gram positive and this as gram negative. Okay. We have added first dye, that dye is what? Crystal violet color is being added, dye is added to both the petri dishes, okay, and then it is washed with alcohol, then treated with a mordant, that is iodine, then treated with a mordant, that is iodine, so that after washing it off the culture will settle back, mordant is added. After that again it is restained, both of them will lose the color, okay. If you counter stain it with one more dye, that is safranin, with one more dye if you counter stain it, one set of bacteria will not get stained, it will not get stained with safranin, it will retain the same color, it will retain the same color, whereas the other set of bacteria will respond to the dye, it cannot retain the stain. That is because of peptidoglycan composition. For example, for your simple example, let me tell you, take a cotton cloth and a silk cloth, keep it in dye, okay, wash it thoroughly, after which if you observe those two cloths, which cloth will retain the dye still? Stains will not be easily removed on what? Cotton cloth. That is because of tight strands, tight and tough strands, cellulosic strands that are present. Same is the case here, okay. Gram positive bacteria, because of more amount of peptidoglycan, it is absorbing crystal violet, so that it is retaining the color even after staining it with safranin. Even after staining this with safranin, it is retaining the same color, okay, because it has high amount of peptidoglycan, whereas the second set it is not the same. It has less amount of peptidoglycan where it can be easily washed and it can be restained again. Got it? So, here if we observe gram positive and gram negative bacteria, major differences is here 80% peptidoglycan, here 20% peptidoglycan. Gram positive bacteria is composed of murin. I told you, right, peptidoglycan, peptido means proteins which consist of amino acids, glucan, glucose units, those glucose units are present in the form of NAG units, N-acetyl glucosamine units, that is known as murin. Whereas gram negative bacteria consists of pseudomurin. Gram positive bacteria, it also consists of teichoic acids. Here teichoic acids are absent. Examples, few of the examples if we have to take for gram positive includes bacillus, Clostridium, Corynebacterium. Okay. As gram negative bacteria can change, can adapt, it is restaining all the time, right? It can change and adapt. Most of the pathogenic bacteria are of gram negative bacteria only. Okay. Most of the gram negative bacteria are pathogenic. Got it? Examples here you can take. Examples here include Salmonella, Azotobacter, Acetobacter, E. coli, and yeah, that's all you can take. Salmonella, Acetobacter, E. coli, Rhizobium, which helps in nitrogen fixation. Okay, you can take the examples. From online, students, you can take the screenshots. Corynebacterium. This is E. Done, ma. Take your time, no problem. Take down slowly. Done? Everything is covered. Membrane bound organelles, genetic material, motile, non-motile, gram positive and gram negative bacteria. Everything is being covered. Now let us look at the key differences between prokaryotic cell and cell. Prokaryotic cell, if you the outermost layer will either be or cell has cell envelope which is of how many layered? 3 layered. Outermost is glycocalyx. It can be in the form of capsule like this or slime layer. Second is what? Cell wall, and third layer is second. In prokaryotic cell, plasma membrane will have extensions called as mesosome. Membrane will also have ribosomes attached to it. Whereas in eukaryotic cell, you can see ribosomes either over the endoplasmic reticulum or inside cytoplasm or inside mitochondria and chloroplast. And you cannot see any extensions of plasma membrane in a eukaryotic. Second difference. And the third difference, though we are discussing at third point, that is the primary difference between prokaryotic and eukaryotic. That is nucleus. Very good, Skanda. What is that? Genetic material is surrounded by a double layered nuclear envelope in a eukaryotic cell. In a prokaryotic cell, genetic material is not completely surrounded by a nuclear membrane. It is placed at an area, but then it is not completely surrounded by nuclear membrane. This is known as what? Nucleoid, and this is known as what? Nucleus. That is the third difference. And fourth difference, if we have to see, membrane bound organelles. Membrane bound organelles, single, double membrane bound organelles, you can see only in the eukaryotic cell. Prokaryotic cell will have, they just have non-membrane bound organelles only in a prokaryotic cell. What are those non-membrane bound organelles? Ribosomes and inclusion bodies. Small questions from ribosomes. What are ribosomes composed of? Ribosomes composed of, good, RNA and proteins. RNA and proteins. How many subunits it have? 2 subunits, larger and smaller. 70S will be having larger subunit as 50 and smaller as 30S. 80S ribosomes will be having larger subunit as 60S, smaller as 40S. Okay, now next, The ribosomes will be in polysome condition in which prokaryotic cells. In prokaryotic cell you can see polysome where to a single mRNA many ribosomes are attached. That is one more difference. Whereas eukaryotic cell will have only single mRNA attached with a single ribosome, which is a monosome condition here. Here it is a polysome condition. Clear? Understood? Yeah, these are the key differences between prokaryotic and eukaryotic. Even if you get diagram based question on the differences also, you should be in a position to answer. Right, ma? Now, look at this tabulated form which I have given completely, which addresses all the differences between prokaryotic and eukaryotic cells. So, here if you can see, first is what? Nucleus. In prokaryotic cell there is no true nucleus, only nucleoid region is present, whereas eukaryotic cell will have true nucleus with nuclear envelope. Second, DNA structure. DNA structure in a prokaryotic cell is double stranded circular DNA. Circular DNA. Eukaryotes, it is mostly double stranded linear DNA. You tell me, ma, DNA is negatively charged or positively charged? Very good. It is negatively charged. To that, some positively charged histone proteins will be aligned with, which will help in condensation of DNA. That we have learnt already in Mol Bio last year. Okay. So, that histone proteins are present only in a eukaryotic cell. Histone proteins are not there in a prokaryotic cell. You have non-histone proteins called as polyamines. You can take down parallelly. Non-histone proteins known as what? Polyamines. Those are present in a prokaryotic cell. Next, cell size is small for prokaryotes, larger for eukaryotes. Membrane bound organelles absent here, they are present over here, both double membrane bound and single membrane bound organelles. Got it? And then cell division is by binary fission, which will not allow, which will not have any spindle apparatus, formation. Binary fission is also known as amitosis, wherein a single cell to divide into two cytokinesis, after that karyokinesis is. Binary fission is also known as what? Amitosis, whereas mitosis will take place in eukaryotic cell. Clear, right? Yeah. And then ribosome type. Ribosome is 70S in prokaryotes, where larger subunit and smaller subunit, here it is 50S and 30S. Over endoplasmic reticulum or inside cytoplasm of eukaryotic cell, you will see 80S ribosomes. For 80S ribosomes, the larger subunit is 60S, smaller subunit is 40S. Okay. 70S ribosomes are also present, but they are present in either mitochondria and chloroplast. They are present in mitochondria and chloroplast. Got it right, ma? And then cell wall composition. Cell wall composition is peptidoglycan here in bacteria. Here it is cellulose in plants. In plants it is cellulose, hemicellulose, pectins and proteins. In fungi it is chitin, whereas in animals cell wall is absent. Examples include bacteria and archaebacteria. Here for eukaryotic cell you can take any members from Protista to Animalia. Next, plasma membrane doesn't have any sterols. For prokaryotic cell plasma membrane you just have those extensions, it doesn't have any sterols. Sterols are absent. Whereas in eukaryotic cell you can see cholesterol. Sterols are present, steroids are present in eukaryotic cell membrane. And respiratory enzymes are released from the cell membrane. from mesosomal sacs, whereas here in mitochondria, aerobic respiration happens. Here entire process happens in cytoplasm. If it is aerobic respiration, it will be adjoined with cell membrane, that happens via mesosomes. Okay, these are the key differences. You can take down, take 2 minutes of time and take down. Students from online, you can have a screenshot. If you are having any doubts, you can ask through comment box, chat box. Is it done, ma? You can make out the differences from diagram also, no? Yeah, done, right? Yeah. Now, let us look at the eukaryotic cell. So, first we will look at the diagrams itself, both for the plant cell and animal cell. Yeah. First, plant cell. Even though the diagram based questions are asked, we should be comfortable in answering them. Outermost layer is what? Cell wall. Cell wall is connected via plasmodesmatal connections. This plasmodesmata will have desmotubulin inside it. Okay, I will tell you in detail when we are discussing about each part of each region of the cell. As of now, you just remember on the labelings what we are learning. After cell wall, we have what? Cell membrane or plasma membrane. I hope you are clear with the synonyms: cell membrane, plasma membrane, selectively permeable membrane or semi-permeable membrane. One and the same. Okay. And then cytoplasm, which is the main arena of all cellular activities. In a prokaryotic cell, though, we don't have any membrane bound organelles. We don't have any membrane bound organelle. So there is no active compartmentalization in the prokaryotic cell. But then in eukaryotic cell, we have single membrane bound and double membrane bound organelles, which gives active compartmentalization of cytoplasma. Cytoplasm is actively compartmentalized with single and double membrane bound organelles. So here if you observe first, non-membrane bound organelle in plant cell is only single organelle, that is ribosome. That is the only non-membrane bound organelle you can see. Single membrane bound organelles include first endoplasmic reticulum, which are the extensions of nuclear envelope. First is rough endoplasmic reticulum and then smooth endoplasmic reticulum. After which you have Golgi bodies or Golgi complex, Golgi apparatus, again single membrane bound organelle here, and these are secreting out lysosomes from the trans phase. Okay, lysosome again a single membrane bound organelle. Vacuole, which is single membrane bound and occupies 90% of plant cell volume. Okay, this vacuole, outermost boundary is what? Tonoplast. The membrane of vacuole is tonoplast. Vacuole is again single membrane bound. after which we have micro bodies like peroxisomes, wherein we come across this term in photorespiration when we are learning C2 cycle and photosynthesis. So, this is also single membrane bound organelle. Clear with all the labelings of single membrane bound? Now, double membrane bound organelles include microtubules, which are also single membrane bound, helps in intracellular transport and it will also help to make up the cytoskeleton. Okay? And then double membrane bound organelles include nucleus. Nucleus we have 4 components. One is nucleolus, nuclear envelope, nucleoplasm, nuclear membrane, 4 components, of which 2 are given in this diagram. Okay? And then mitochondria, which is mostly sausage shaped, sausage or cylindrical shaped double membrane bound organelle, after which we have chloroplast. Okay. Now, we can comfortably answer the diagram based also, no, if it is asked from a plant cell. And then this first formed layer is what? Middle lamella. Middle lamella in a matured plant cell, if the labeling is on the outer side, no, that is middle lamella only. Labeling is on inner side, then it will be secondary cell wall or primary cell wall. Got it right now? Clear with the plant cell? Next, animal cell. Here, animal cell, this diagram is from some intestinal cell because we can make it out because it has microvilli which actively take part in the absorption of minerals, food nutrients and all. The outermost limiting boundary is what? Plasma membrane. First, coming to non-membrane bound organelles. Plant cell will have only ribosomes, whereas animal cell will also have centrioles, 2 cylindrical structures perpendicular to each other. Okay? Centrioles are again non-membrane bound organelles, centrioles and ribosomes. Here also you can see cytoplasm is compartmentalized with all membrane bound organelles, and single membrane bound organelles include smooth endoplasmic reticulum, rough endoplasmic reticulum, Golgi bodies or Golgi apparatus and lysosomes. These all are single membrane bound organelles, and double membrane bound organelles include mitochondria and nucleus, that is all. Peroxisomes are also here, which are single membrane. See the labeling, just remember and register the labelings in your mind. And in nucleus, 2 labelings are given, nucleolus and nuclear envelope. Nuclear envelope. Clear till here, right? Any doubts anywhere? Yeah. And then, so these are the basic differences between a plant cell and animal cell. We will look at the differences and then we will move on to the cell wall and cell membrane. First, plant cell outermost is cell wall, which is made up of cellulose, hemicellulose, pectins and proteins. Animal cell wall is absent. Here the shape is regular or rectangular. This shape you can ignore, nobody will ask you. Plastids are present. We have 3 types of plastids. Can you remember and name it? What are 3 types of plastids? Chloroplast, chromoplast, leucoplast. Very good. Chloroplast, chromoplast and leucoplast. 3 types of plastids are present in a plant cell. They are absent in animal cell. Vacuoles are very small or limited in an animal cell, whereas it is large. It can form large central vacuole, also known as sap vacuole, which occupies how much percent of plant cell? 90%. Centrioles are absent in the plant cell. Centrioles are present in the animal cell. What is the function of centriole? Centrioles function, if you remember, they will help in the formation of spindle fibers. Spindle fibers play key role in cell cycle and cell division especially. Spindle fibers will play key role in cell division, and for the formation of spindle apparatus or spindle fibers, centriole play crucial part. In animal cell they are present, in plant cell they are absent. Then how the formation of spindle fibers or how the function is compensated? That is by microtubular organizing complex in a plant cell. In a plant cell it has what? Microtubular organizing complex. This is present in the plant cell. Got it? With this spindle apparatus is formed and the function is compensated. And then lysosomes, they are rare or less present in a plant cell, they are present in abundance in an animal cell. It is autotrophic mode of nutrition, heterotrophic mode of nutrition. You can say holophytic and holozoic as well. Cilia and flagella are absent, they are present here. Cytokinesis is via cell plate formation, which is centrifugal in plant cell, whereas animal cell it forms via cleavage furrow from periphery to center. This type of cell plate formation or cell cytokinesis is centripetal. This is centrifugal and that is centripetal. Stored food material in a plant cell is starch, stored food material in animal cell is glycogen. Got it? So, these are the basic differences, 80% of which you already know. Few points you have to add on is about microtubular organizing complex that is present in a plant cell. Instead of centrioles it has what? MTOC, microtubular organizing complex. And few points to add on about lysosomes, they are less present in a plant cell, more in an animal cell. Third you can add on is the centrifugal mode of cell plate formation in a plant cell, centripetal mode of cleavage furrow formation in an animal cell. Take your time and note down. Add on points if you are taking also, it is fine. Done, right? Yeah. Now, cell wall. So, before learning about cell wall actually, once let us recall that two kingdom system of classification, right? Two kingdom system of classification. Who gave us two kingdom system of classification? Carolus Linnaeus has given us two kingdom system of classification, where he has classified the entire living organisms into two kingdoms. What are they? Plant kingdom and animal kingdom. Into two kingdoms he has classified. All the sort of living organisms, be it prokaryotic, eukaryotic, unicellular, multicellular, autotrophic, heterotrophic, he has classified. He has many criteria, of which cell wall is the major criteria he has taken. What is the major criteria? Cell wall. Cell wall is the major criteria for two kingdom system of classification, which is proposed by Carolus Linnaeus, wherein he has classified into two kingdoms. What are them? Plantae and Animalia. Plant kingdom, animal kingdom, right? So, be it prokaryotic or eukaryotic, unicellular, multicellular, photosynthetic or non-photosynthetic, if they have cell wall, so he has placed it in plant kingdom. If they have cell wall, he has placed it in plant kingdom. Likewise, bacteria has landed into plant kingdom, yes or no? Does it have cell wall or not? It has cell wall, made up of peptidoglycan. Fungi is placed into plant kingdom only, though its nutrition is heterotrophic, because it has chitinaceous cell wall. Algae is placed into plant kingdom. Unicellular, multicellular, all bryo, pterido, everything, if they have cell wall, they are placed into plant kingdom. If they doesn't have, they are placed into animal kingdom. Likewise, fungal cell wall you have what? Chitin. Fungal cell wall is made up of chitin. Chitin is also known as fungal cellulose. Chitin is also known as fungal cellulose. It is a polymer of NAG units, which are N-acetyl glucosamine units. It has N-acetyl glucosamine units. Second is what? Algae. Second is algae. Algae consists of, algal cell wall consists of galactans, mannans and minerals like calcium carbonate. Galactans, mannans and minerals like calcium carbonates. Plant cell wall is composed of insoluble polysaccharide, that is cellulose. Cellulose is a homopolymer or heteropolymer? Homopolymer of glucose. Cellulose is a homopolymer of glucose. Okay. It consists of cellulose, hemicellulose, pectins and proteins. Composition is very important, ma. We have recalled two kingdom, plant and animal kingdom. In plant kingdom, bacteria is placed because it has peptidoglycan. Fungi has what cell wall? Chitinaceous cell wall, and it is also addressed as fungal cellulose. It has what units? NAG units, which is N-acetyl glucosamine units. Algal cell wall is composed of galactans, mannans and minerals like calcium carbonate. Plant cell wall is composed of insoluble polysaccharides like cellulose, hemicellulose, pectins and proteins. Got it? Clear all? Now we will see on how a plant cell wall is formed. To learn clearly about plant cell wall, we have to learn from plant cell cytokinesis. Okay, listen carefully everyone, sit straight all, listen carefully, drop your pens. So, if this is the region at the end of telophase, what are the 4 stages of karyokinesis if you remember? 4 stages of karyokinesis: prophase, metaphase, anaphase, telophase. Telophase is the last stage, where you can see the genetic material and nucleolus aligned to 2 opposite poles, right. In a plant cell, what happens is, at the center phragmoplast is synthesized. What is it, ma? Phragmoplast. The center phragmoplast is synthesized. This phragmoplast is synthesized from what? From Golgi bodies, and it is rich in calcium pectate and magnesium pectate. Magnesium pectate. This phragmoplast from the center, it will start moving to the periphery, and this forms into a cell plate. Okay, this forms into a cell plate. Cell plate will further extend to the corners and completely it will form the cell wall. First formed cell wall is middle lamella. This is the first formed cell wall. Got it? A quick recap for you people. Initially, after telophase, which is the last stage of karyokinesis, at the center what is synthesized? Phragmoplast, that is being synthesized from Golgi bodies, and it is rich in calcium pectate and magnesium pectate. This phragmoplast will slowly move to the corners, forming up what? Cell plate. So, the movement is from center to periphery. Any movement which is from center to peripheral ends is known as what? Centrifugal pattern of cell wall formation. In plants it is what pattern? Centrifugal pattern of cell wall formation, and at the end it will move to two opposite poles and it forms the first formed middle lamella layer. This pattern is centrifugal pattern of cell wall formation. Clear, right? So, in this diagram can you see at the center what is first formed? Middle lamella. So, how middle lamella is formed, you have a clear idea. Now, let us see how primary and secondary cell wall layers are formed. Okay? Primary and secondary cell wall layers. Assume this as the cell, plant cell. Okay. First formed layer is what? Middle lamella, right? This is the common wall layer between two plant cells. First formed layer is middle lamella, which is common wall between two plant cells. Okay. Next, inner to the middle lamella, primary cell wall is formed. Primary cell wall is not formed outer to the middle lamella, inner to the middle lamella it is formed here. This is second formed layer is what? Primary cell wall. This is the second formed layer which is inner to the middle lamella. Got it, ma? Primary cell wall is single layered. It is single layered. It is thin and elastic. Okay, extensible, made up of parenchyma. Got it? Primary cell wall which is inner to the middle lamella, it is single layered, it is thin, elastic, extensible, and it is made up of parenchyma, loosely arranged. Inner to the primary cell wall we have secondary cell wall layers. Secondary cell wall layers can be 2 or 3 layered. It can be 2 or 3 layered secondary cell walls. Okay, here you can see the clear differentiation. Third is secondary cell wall. It is 2 to 3 layered with suberin, lignin and pectin depositions. Got it, right? So, the common wall layer is middle lamella. Next to middle lamella what is formed? Primary cell wall which is thin, extensible, made up of parenchyma. Inner to the primary cell wall, secondary cell wall is formed. Secondary cell wall is how many layered? 2 to 3 layered. The secondary cell wall will form in tall plants like gymnosperms, angiosperms. Not all plants will have, not all regions of the plant also will have secondary cell wall layer. But then if it is forming, it can have 2 to 3 layers. It is made up of suberin, lignin or pectin depositions. So, as the cell is increasing in its size, as cell is growing, secondary cell wall layers are growing. The primary cell wall layer which is thin, right, it can break, it can lose its structural integrity. Primary cell wall can lose its structural integrity with the growth of the cell and with the increase of secondary cell wall layers. Primary cell wall can lose its structural integrity. It can have, damage can happen to this primary cell wall. Got it? Now, it is important to know the cell wall layers from out to in and in to out. Listen, this is the center of the cell. Assume cytoplasm. Okay. Closer to the cytoplasm, what is present? Cell membrane. Next to cell membrane, secondary cell wall is present. Next to secondary cell wall, primary cell wall is present, and then middle lamella. From out to in, if we see, middle lamella, primary cell wall, secondary cell wall, cell membrane. This will be the hierarchy. Take down more. Any doubts you are having on formation of cell wall? No, right? Here also compositions of cell wall. From cell wall, what you have to be clear on? Compositions of different cell wall, bacteria, fungi, plant and algae, and then you have to know on formation of cell wall, and then you have to learn on primary, secondary cell wall layers, how they are being formed in a plant cell. Yeah, take down. Tomorrow we will start on cell membrane.